All right, so in this short lecture, we're going to discuss mitosis and briefly introduce meiosis. Okay, so we have the cell cycle is divided into two main phases. So you have interphase and mitosis, and there are subphases within each of them. So for interphase, you have G1, S phase, and G2. Once interphase is complete, we then move on to mitosis. With mitosis, we have prophase, metaphase, anaphase, telophase, and cytokinesis. So the easy way to remember this is just PMAT for mitosis. Okay? All right, so that's for the cell cycle and all the different phases. So on what I have these two drawing boards here. So let's go ahead and identify some of the different parts of it, because we want to distinguish between meiosis and mitosis. So we start here and we end here for mitosis. The product of mitosis is this. We have two identical diploid cells. Okay, so we have two identical diploid cells, and for meiosis, we have 4 haploid cells. Okay, so let's write that on the board. So for mitosis, we have 2 identical daughter cells. And these 2 identical daughter cells, they are diploid. And we can write 2N. For meiosis, we have 4, there's some variation. So variation cells, varied cells, write that. Varied cells, and we're going to say we have, these are all haploid. So this is N. So now I want to explain to you what is the difference, like what does diploid versus haploid mean? Okay. So what is the total number of chromosomes that we have? Does anyone know? 46, yeah, so we have 46 total chromosomes, and we have 23 pairs of chromosomes. And so the reason that we say that they are pairs is because we get 1 from mom and 1 from dad. Okay? So that's why there are 23 pairs and a total of 46 chromosomes. Okay? So when I say it's diploid versus haploid, how many total chromosomes are found in the cells after mitosis? How many total chromosomes? So you just multiply 2 times N. So N is equal to 23. So there's a total of 46 chromosomes in these cells. Compared to meiosis, there's only 23 chromosomes here. Okay? So why is that the case? Well, so for this, so if you play a little bit of Marvin Gaye, right, having some wine or everything, what can happen? Fertilization, right? So this, this is when this haploid gamete will fertilize the egg. Okay, so here's a fun fact or something interesting to know. So in one male ejaculation, there is over 1 billion sperm that are released in one ejaculation, compared to a woman, which is 1 egg each month. So there is a huge difference between the two, right? So if you're ever feeling down, just remember, you're a winner. You were the one that fertilized that egg, okay? All right, so that's just to kind of like get some perspective, because it's really amazing, like how many and the whole process. But we'll learn all about reproductive, right? That's all for next semester. But we're trying to relate this to, comparing mitosis versus meiosis. Okay, so this is the reason why you go have two haploid cells that become diploid, because one haploid cell from mom, one haploid cell from dad, and you form you, and you get our diploid cell. Make sense? Yeah? Okay, cool. So let's go back and go back to the board here and finish the different phases that we're in. So what is this? Interphase. This, both of these are prophase. This is early, late. Where they line down the middle? Metaphase. After metaphase, we have both of these are in the same. Anaphase. So early and late. This is telophase and the completion of cytokinesis. Okay. So one thing is that with cytokinesis, it starts here in late anaphase. And it ends here. So the difference between, so mitosis is the division of the nucleus. Cytokinesis is the division of the, so not cyto, I'm going to put cell membrane. The division of the cell membrane. So you can see how on number 6, right, that's when this cell membrane, it's starting to divide. This is when it starts here in late anaphase. And then it's complete after telophase. Okay? All right, so that's the difference between the two here. Okay, so we talked about mitosis and how we get diploid cells. But now let's go back here to the front, to the very beginning of interphase. So during interphase, this loosely coiled DNA, it's known as chromatin. So you have chromatin and then you have chromosomes. So chromosomes are the condensed version of it. Okay, so another thing I want to point out, so how is it that you're able to have chromatin versus chromosomes? How is it in the loose, so loosely, so it's kind of loose, or it looks like spaghetti. Okay, so spaghetti, and then chromosomes are condensed. Condensed. So the reason we're able to do this, go from this form to this form, is because DNA will wrap around, so let me draw a strand here. So DNA will wrap around this protein here. So this protein right here, this is known as a histone. So whenever we're in the condensed form, when chromosomes are in the condensed form, it's going to be wrapped around really tight. But whenever you're in interphase, when it's chromatin, they're not going to be tightly held on those histone proteins. Okay, so it's just like if you think about with gene expression. So DNA contains certain genes, and from there we can make mRNA, we can make transcripts, right? And so in order for that to happen, it has to unravel so you can read that sequence and then make our transcripts and then get it out to the cytosol. Okay, so that's the important role here of the histone proteins. Okay, so everyone good on the difference between chromatin versus chromosomes? All right, so once we get into prophase, we have our condensed chromosomes, and interphase we have chromatin. All right. So chromatin, and then all of these are chromosomes. And let's look at, so it's going to be a little bit difficult here to see. Let me see if I can get closer towards the camera. Okay, so let's kind of like identify some of the different components here within the interface. So we have, this is the plasma membrane here on the outside, and then number 3 here, this is the what? The nucleus. So you have the nucleus, and then number 4, this is the outer part right here. So this is the nuclear envelope. So you have the nuclear envelope, which houses all of the DNA, and then this little green dot right here, this green structure, this is the nucleolus. So the nucleolus is where ribosomes are manufactured. And then here on the outside, this is the cytoplasm. So you have the cytoplasm, and then both of these, so you have the centrosome, and then the centrioles are these ones right here. Okay, so that's the components for number 1. Moving on to number 2. We now have our condensed chromosomes. And so this part here in the center, that's known as the centromere. So you have the centromere, and then you have what's also found here is what's known as the kinetochore. So once we get to, once we get to metaphase, they're lined up down the middle, and so these kinetochore microtubules, that's what's going to pull the chromosomes towards the opposite ends to start anaphase. Okay, that's what they attach to. They attach to the kinetochore. That's why they're called kinetochore microtubules. But if we look up here earlier, this is just the mitotic spindle that will develop into this part. Okay. All right. So what is the relationship between these two? So once we duplicate that chromosome, we then have what's known as sister chromatids. Okay, so they're sisters. So these are sister chromatids. And then number 9, nothing really here, just the mitotic spindle. I've already discussed this one. And there's no details in these two. Both of these are pretty much the same as what was here at the top. Nucleus, nuclear envelope, nucleolus, and then also here on the outsides as well. Okay, so pretty much the same. The other thing that I wanted to point out is that, so if you look at late anaphase here compared to telophase, this is how I was able to tell the difference between the two. So if you look at this one, you'll see that the nucleolus has reformed. It's already there. Compared to here in anaphase, the nucleolus is not there. Or the nuclear envelope, the nuclear membrane that surrounds the nucleus, okay? So it's not there. All right, so any questions regarding the mitosis board? No. Okay. So the last thing that I wanted to discuss with you, so I mentioned that the kinetochore microtubules, that's what attaches to the centromere. So that's what attaches to the centromere, which is on the chromosome. But I want to talk a little bit about microtubules, okay? Because microtubules are a component of the cytoskeleton. So there's other various types of proteins here for the cytoskeleton, but we're going to focus on microtubules specifically because that's what is helping to pull the chromosomes apart. All right, so microtubules are composed of a protein that's known as tubulin. And so for tubulin, you have the alpha tubulin and then you have the beta tubulin. So what will happen is you'll have the alpha subunit there, and then you'll also have the beta subunit there. So there's these, both of these two different proteins, you consider them isotypes, that are very similar together, and they help to form our microtubules, okay? And so this whenever we have two different subunits that form together, that's how we make up what's known as the quaternary structure. So that's over here. So this is what our quaternary structure, when we have multiple proteins that have completely folded and they're in their native structure, okay? But there's a process for us to get to this point, okay, for the folding of proteins and how they become their mature structure, okay? So let me ask you this. So what are proteins composed of? Proteins are polymers that have individual monomers. What are these monomers called? Amino acids. So we have our protein here that is composed of amino acids. So we'll have a long chain of them going like this. And the functional difference, like what are the functional groups of here? We have this, and then we have this. So this here is my amine group. This is a carboxylic acid. And then we also have the R group here that's on the amino acid. So when we form a single protein, what's going to hold them together is a region that's known as the peptide bond. So a protein is read from the N terminal to the C terminal. So this is, I'm only going to draw two of them. Just for, I'm going to write this COOH. This is a H, and this is a H, and this is the R group, okay? So I have two different, two amino acids here that are held together by this part in blue. So this part right here, this is known as the peptide bond. All right, so fairly straightforward. It's just a linear sequence. But then what will happen is we'll get to our secondary structure. So our secondary structure, so y'all should know this from functional. We have alpha helices and beta pleated sheets. Ringing a bell? Yeah? So the alpha helix, it'll kind of look like that, but then the beta pleated sheets, they'll kind of look like this in some. So if you think about like curtains, you know, when you have curtains, that are kind of like squished together, that's kind of like the shape that they're forming. Okay, and so what's holding both of these types of bonds or in their structure, we use hydrogen bonds. So hydrogen bonds is what's keeping them together. So the carbonyl, so this part here, let me draw it better. So you'll have the carbonyl, which will bond to the amine group right there. I'm just going to write R group. Okay, so we'll have some hydrogen bonding here for the alpha helices and the beta pleated sheets. So that's moving into the secondary structure. But once we get to the tertiary structure, I mentioned about these R groups. Right? And so these R groups is going to determine how the protein behaves once we get to the tertiary structure. So with hydrogen bonding, right, we get, so we can get our OH here, so hydrogen, so this would be positive, and then I can have my amine group here. So this is an R group, this is an R group. For a salt bridge, so this is whenever we have something positive and negative when they charge together, right? Because there's various types of amino acids, and so this is a carboxylate, and it'll bond here with something that's positively charged. So I have NH3+. So N, and then I'll put N, just put an R. Okay, so that's the formation of our salt bridge. And then we also have our disulfide bond. So that just means we have an S. So it's formed from a thiol bond, but we have two of these sulfur residues that are bound together. And then the other type of interaction will be our hydrophobic interactions, in which they kind of like stack on top of each other. So that's how we reach once we get to the tertiary structure. And so we finally get to our quaternary structure where we form two of our subunits. Okay? So any questions regarding the folding of proteins? No. Okay? Are we good on mitosis, how it's different from meiosis? Yes. Okay? All right, so that's it for this lecture.